cow is 50 minutes. Header I will be showing the 2 minutes left sign. If you don't see it, we're probably going to cough in a microphone or rudely interrupt you for a moment, so try to be on time. First talk is Dr. Marcin Nowick, University of Tennessee, Genomics and Genetics Analysis of Flowering Dogwoods Resistance to Powdery Mildew Fungus. Would you please come in?
One, two, three. Good morning. Wake up, everybody. The wine tasting survivors, hello. So thank you very much for having me, and thank you for this platform to share our results. And the Polish way, I will start from the finish. I am contractually obligated to have you guys ask me questions, otherwise I will have to pay for my trip myself. And let's go to the dogwoods. So this is our organism of focus, beautiful trees, and they are occurring in they naturally grow in North America, and they have many uses. I will get to it in a second. And this is the symptomatic powdery mildew dogwood. And if you want to learn more about our research from other topics related to dogwoods and other beautiful plants, you are kindly invited to follow that link. There we talk about population genetics, evolutionary history, and other cool stuff of dogwoods and many other plants. So again, forest trees that are actually tapping into this industry, right? For beautiful ornamental plants, so landscaping and nurseries and such. In the spring, the bracts can differ in colors from pure white to deep red. And the breeding of dogwoods is an ongoing business, and there are also other traits that are being bred for. The trees also in the natural settings have or environmental importance, and there are many birds and mammals that are actually feeding on dogwoods. So we have deer browsing on the branches, but the fruits, the drupes, are rich in calcium, fat, and other micronutrients that are important for emergency feed. The closely related Asian dogwood, Cornuscusa, is also an important ornamental crop, and this is the picture from one of our breeding sites of Cornuscusa in Oak Ridge, Tennessee. So they can actually be hybridized all together, both together when you synchronize the flowering time. There is a nice phasing of the flowering time which increases the ornamental values of those plants. So for either Cornus florida and Cornus scusa, there are more than 100 commercial cultivars developed, and they are in circulation, and the hybrids amount to approximately same number. And there are about 10 million dogwood trees in sales annually in US, and that value is about, in retail and wholesale, is about $30 million. Roughly half of those trees are being bred in the nurseries located in our state, in Tennessee. So this is bridging between the forestry and industry, again, trying to tap all this great commercial potential. In Europe, we have Cornus mas that is being also consumed by humans. And in following my Polish heritage, Cornus mas can be making also other products. Very noble drink, if you ask me. So symptomatic dogwoods are essentially not as valuable. They can lose their sale values because people don't want to have such a nasty looking thing in their garden. So what happened was that this fungus, Erysife pulchra, was actually, we suspect, we have very strong evidence to claim that this fungus was brought over to North America. And there was a dramatic increase in protection costs. So nurseries normally had to spend about $50 a year per hectare to protect young dogwood seedlings. And after this fungus started being present and started causing problems, these costs skyrocketed to about $2,000 per hectare. So this is a lot of money that the nurseries had to spend on top of everything else. And this fungus is reproducing mainly asexually throughout the history from the first presence in the 1970s, 80s in the US. We started observing decline of the sexual ascospores, and we know that Erysife pulchra and the related species are present throughout many ornamental plants. And we think that Erysife pulchra may have come from the Cornuscusa from somewhere in Asia, possibly, because essentially those plants are much more resistant. They barely show any symptoms of powder mildew. The other related Erysife cornicola, this is by IPS sequences. Erysife cornicola was extracted from the controversa that is also coming from the Asia. So Cornus florida is a tree located to a major part of the eastern US, and Chris Wyman, our previous grad student, analyzed the diversity of this pathogen following main routes in the US, and we had many samples collected. We collected about 150 samples of this obligate parasite, but after analyzing several SSRs, we had to throw away roughly half of those samples because they were clonal in character. So we only wanted to retain the unique multi-locus genotypes. So Chris concluded that this pathogen has a very low genetic diversity, and linkage disequilibrium also spoke for the predominantly asexual reproduction throughout the US, and we finally concluded of lack of definite population structure. So following that, we also developed genomic resources for the Erysife pulchra, and that was published recently as well. So those two studies were linked by the feature of this resource serving to development of those SSRs. This gentleman, Dr. Robert Trujano, is a huge in breeding dogwoods, and he was the one who discovered the natural resistance in a cultivar later called Appalachian Spring, resistant to Discula destructiva, another major pathogen of dogwoods. What they did many years ago was something pretty ingenious. There is a very strong self-incompatibility in dogwoods to the point of not getting any seeds whatsoever if you try to self-pollinate the plants. So they crossed Cherokee Brave with Appalachian Spring. These are two cultivars of dogwoods varying in the ornamental traits, and they develop F1 population, which is pretty straightforward. But then to get the F2, you cannot self-pollinate those plants, right? So we had to go through the pseudo F2, and that essentially was achieved using the honeybee-mediated pollination. And we planted out many of those progeny, and in 2021, we had more than 150 of those plants growing in Oak Ridge. Happening because of, again, self-incompatibility issues, but I want to attract your attention that this F2 population is greatly varying in ornamental traits. We see that there is variation in the leaf color, even though those two originating cultivars did not show any red leaf color. The F2s do show that, and there is nice variation in the bracts color as well, a nice white and red separation. So this was a great resource to follow up. And so they also concluded the same self-incompatibility about the crosses in Cornuscusa and some other species. But this great resource was used towards assessing the inheritance of a leaf color at the early stage. You can do this because you can see the leaf color already in the first, second year. It takes about 5 to 7 years for the plants to start flowering, so they couldn't do it at that stage. But they did figure out that this is a pretty simple inheritance for 255 SSRs on about 94 plants, which is a like nice PCR plateful. they developed a first genetic map for Corns, Florida. And the leaf trait color was located to the linkage group 3 and linkage group 8 with a nice percentage covering the variance. So by now, as I told you, those plants do start flowering, do produce flowers, and we observed separation of those traits, meaning that the leaves and bracts color are independently inherited. So this research is following in the labs, and Jacqueline will tell you more all about it whenever she graduates. She was also the one carrying out the pulchra resistance analysis. So this is at the time when I joined the lab as a postdoc, still in 2016, and we started going in this field and collecting the data on the traits, on the leaf color, on the bracts color, and on the natural symptomatic level of symptoms for the powdery mildew. So you see here in this big table, throughout the years, we have been coding each plant depending on the severity of the symptoms, from very terminal leaves only showing some powdery mildew, through the whole branches, through essentially at least half a plant covered and showing these powdery, really like a meal type symptoms. This varies by year, which is not surprising because it could be very strongly environmentally directed trait. And we have a weather station sitting just next to our experimental field, but this is Oak Ridge. All the data goes down to the Oak Ridge National Lab. So this is a treasure guided and guarded much more than the nuclear bomb secrets. So just last night, irony, I received email from Ed, who is managing the data, giving me access to it. So I'm happy to report on that later to you if anybody's interested. So we picked up 2021 data from pulchra infestation because it followed the natural distribution as one of the few years. And Jacqueline concluded this is a duplicate gene action with 15:1 segregation, and we were able to place QTLs on the chromosome linkage group 3 and linkage group 10 with 14 and almost 13% of variance explained by those QTLs respectively. Moving forward, there were other groups who are also breeding dogwoods, and they also did something similar to us. So there was a group in State University of Tennessee in essentially collaborating with us. They developed their own 2 crosses, but they had different starting materials. So they could not use entirety of our 255 markers for the maps because they did not have the differences, they didn't have the differential responses from the parental lines. And so they concluded that in their materials, in their F2, they had QTLs located at groups 4, 5, and 9. And just this year, earlier this year, Thomas Molnar group from Rutgers published F1 genetic map based on SNPs, and that was, on the other hand, using a baseline of the dogwood draft genome from 2017. And in their research, in the F1 population, they had one strong QTL located in their linkage group 3 that corresponded to our chromosome 5. So we're switching gears. We have more people and we have more sophisticated tools, luckily. And Lav Yadav, who is a postdoc in McStadden lab, and Trinity Ham, a PhD student from the same lab, started doing very cool stuff. So Lav pushed for the flow cytometry analysis of the Cherokee Brave because our partners are doing Appalachian Spring, so we are focusing on the other parental line. And we got this haplotype, this 1X size for the genome. Whereas Trinity was working with the long molecule sequencing. She used the data from the Hi-C and the Hi-Fi. Hi-C is helpful for the establishing the haplotype blocks, and the Hi-Fi reads by circular sequencing ensure very high accuracy of the reads. So in the first run, she got 471 scaffold with this total basis. But then, after scaffolding with juicer and 3DNA, she was able to achieve 11 pseudomolecules, 11 pseudochromosomes, which is pretty cool, and this is a substantial improvement compared to the 2017 draft genome. So we left her working on all this data. This picture was taken just one day before we left to Lisbon, and poor thing is working very hard. We altogether got about 4,000 SNPs between those two parental lines, so it shows you that there is not as much underlying differences between those two lines, but enough for us to be meaningful and to be helpful. And we also narrowed it down to several hundreds of high-quality SNPs to guide the following analysis. So we can foresee several ways of going forward, either combining with the 94 individuals for SSRs or just analyzing by the SNPs, but we want to also leverage the available RNA-seq resources that are in the NCBI. So with that, I want to thank you for your attention and asking for questions later.
Thank you so much for the talk. Speaking about genetic, this is wrong, genetic copy.
Thank you, thanks. Good morning, everyone. So what I'll be talking about today is a system that we've been studying for quite a few years already, and the work we're doing and that I'll be talking about today is trying to understand the evolutionary basis of these resistance traits that we've been studying in spruce trees. I'd like to start by acknowledging the main collaborators on this work. So I started this when I was in Canada at Laval University, and Geneviève Parent was my postdoc there, and Isabelle did a lot of the lab work on the project. I'll also be talking about earlier results. A lot of them were developed by Melissa Maidroy, who was at UBC at the time, is now in Norway. And this is work. Yeah, okay. Our present team at Oxford, I'll be talking about a new project that we're launching. So this is the main study system that we've been working with. So white spruce is a transcontinental species that goes all across of Canada. And the spruce budworm is a small moth that also has quite a wide distribution, largely overlapping that of white spruce. White spruce is one of its main hosts, but not the only host. It also attacks other spruces and balsam fir. We studied a number of populations that are located in the east, and the 3 dots are simply illustrating different provenance trials, progeny trials that we worked with. Think of those as common garden experiments. So to illustrate, maybe a bit of background. So spruce budworm is a massive problem in Canada, and this is just to illustrate an overview of Canadian forests. So if the whole section is all of Canadian forests, this is the amount that's harvested every year, the amount that's lost to fires, and the amount that's lost to insects. And the spruce budworm alone is responsible for around 1/3 of that loss. So it's a massive issue, and it's been one for a very long time. Is attacked by this budworm, but colleagues in Canada have discovered that some trees are much more severely attacked than others. And in this study on the far right, you'll see a tree that's basically resistant. Here, resistant means that there's no more than 20% defoliation. So the spruce budworm eats the needle. So it's not purely resistant, it has a high level of resistance. On the left, you see a tree that was quite severely damaged, but is recovering, so we're calling it susceptible. It lost up to 70% of its foliage, and some other trees died following even higher levels of attack, like this tree here. So for presentation purposes, I'll refer to resistant and susceptible, but remember it's a quantitative variation. And what we've learned is that the resistant trees accumulate quite high levels of these two acetophenones. So as you can see, these are phenolic compounds, piceol and pungenol. And both resistant and susceptible trees have the precursor, which is glycosylated. So picein and pungenin, these are present at even higher levels in both susceptible and resistant trees. And we've identified the gene responsible for this step through transcriptome profiling, and we know that there's a very large difference in transcript levels in resistant and susceptible trees. It's constitutive, it's observed at different times during the year, and the two acetophenones are also quite different, and whereas the picein doesn't vary much. Now I'd like to make a couple of comments. You may be thinking, oh, that's not a big difference in transcript levels. Note the scale. This is a log scale. So we're talking about 3 orders of magnitude here. 1,000-fold difference in transcript levels. The levels of accumulation of these compounds is 5% to 10% of dry mass. It's quite massive. The precursors are even at higher levels, so up to 25%. So very abundant compounds. We published a review paper about 3 years ago, but some of the initial discoveries date back a little further. And it's important to note that gene expression of this beta-glucosidase 1 gene correlates with the accumulation of these compounds. Now it correlates, it doesn't explain it entirely. We also were able to test the effect of these compounds on spruce budworm development. It's a tricky business. These insects are incredibly small. We're applying them with a paintbrush to the buds and looking for defoliation and then looking at development of the insects. In general, development is reduced and delayed. The only statistical result in this study was that the females were smaller in the when placed on resistant trees. So here, NR is for non-resistant. Generally, the males were also a bit smaller, but the wasn't statistically significant. These phenotypes. Because of inherent variation. So we're doing this on trees in the field. So a bit more on the genetics. There's a lot of variability in the population when you look at these resistance biomarkers. From left to right, you have the picein, which is present in both susceptible and resistant. This is looking at 3 different provenance trials, 3 different progeny trials, different populations, the piceol, and then the transcripts. And you can see the distributions are quite different. When it comes to the defense compounds themselves, a lot of trees have very low levels, but then quite a few of them have these increased levels. The distribution of RNA transcripts is quite different, with some trees having very high levels, but many having quite low levels. So, a few other points. We know it's constitutive, we know it's under strong additive genetic control, highly heritable, and these results are being used by tree breeders. We also carried out a GWAS analysis, identified around 15 loci that explain 20 to 40% of the phenotypic. We detected hotspots of variation across the landscape. So if you look at the origin of the progeny we studied, some populations have higher levels of these biomarkers than others. It's not a straightforward case, but we do detect a statistically significant signal. What we don't have an explanation for is the molecular basis of the variation in gene expression. And we've tested a few different hypotheses, and the one I'll be talking about today is copy number variation. Now, I'll tell you right away that we can't explain the phenotypic variation with copy number variation to our satisfaction, and we need to continue working on that. But we detect some very striking results when it comes to copy numbers. So we carried out a targeted analysis of copy number on this gene family, and first of all, we developed a system in which we synthesized a longmer of the beta-glucosidase 1 gene so that we could develop standard curves for quantification. And if you look at the two graphs on the left, the results from the longmer are the black dots. And what we're doing is we're taking this longmer and we're making serial dilutions and we're carrying out our quantitative PCR. And then the white and the gray are results of serial dilutions of genomic DNA for single copy genes. So we know that we're able to use our longmer in a way that very closely mimics what we would expect for a single copy gene in the genome. On the right, what we're seeing is in the black dots here, it's slightly reversed, is what we detect for the beta-glucosidase gene. So these are CP values from our TPCR, a very simple presentation. You see it shifted to the left by quite a few positions, and that indicates that there's many more copies in the genome. So that's kind of the first evidence that there's copy number variation. But it's not quite that simple. It turns out that we're able to detect also in targeted genome assemblies several different forms of the gene, and they're illustrated here with exon and intron structures and different insertions and deletions in the different forms. And the little black bars, as shown here, represent variation. So we're positioning these in ways where we are only amplifying genomic DNA. It always spans an intron-exon junction, and we're using variations between the forms to make the amplification specific. So what did we find? So forms 1 to 6 are in the center of the graph, and you can see that we have copy numbers that reach up into the several hundreds for individual forms, and in some cases we're looking at thousands of copies. If we add up all of these results, we get a sum, and this is around 1,500 is the median. But some individuals have many more hundreds of copies, so above 4,000 and up to 6,000 in a few cases. If we use primers that are non-specific, that amplify any of these forms, the numbers are even a little higher, indicating that we're missing some of the genes, and there are more in the genome. We looked at inheritance of the copy numbers across 9 full SIB families for these 6 different forms. What you would expect would be between offspring on the left and parent on the bottom. Okay, you would expect a one-to-one relationship. We have some deviation from that. So where are we going next with this? We're setting up a landscape analysis. We have a couple of challenges. Finding more sequences in the genome is still quite tricky. But we have an opportunity where we know these compounds are present not only in Picea glauca, white spruce, but also in Norway spruce. And so what we've decided to do is set up range-wide studies in both those species in collaboration with Nadir and Aris Janssens at Silava. We've carried out the sampling this last July. We have several hundred samples to analyze now, and we have a number of objectives: improving the gene models and improving our CNV detection methods so that it's a bit more diagnostic, and then looking for associations with environment and natural enemies. So we're using targeted sequencing to do this with biotinylated probes. Interestingly, there are many species of spruce budworm. I only talked about one, but when you look at across the landscape, there are several species. So does that drive any differentiation between populations? We have preliminary results from our PCR method for Norway spruce at the bottom here, showing that there is copy number variation, but when we use the non-specific probes, the numbers are much larger than the sum here. So again, for Norway spruce, we need to improve our gene models. And I just want to finish up with one more result. My time is over. So we've looked at this gene. We've also identified this gene. And with our new targeted analysis approach, we show that we're detecting thousands of copies for the beta-glucosidase gene and only much smaller numbers of copies for this other gene. So it validates our findings. So from now we're moving on to carry out this larger scale analysis, and the relationship to phenotype still remains our most complex question to address. Thank you for your time.
Next we have of nemacidal activity of fungus to pine wilt disease.
She's not coming.
She's not coming? So we've got you instead. Right. Mouse? I'll do that. Thank you.
Well, hi again. We are sorry, but Lucia is not coming today, or at least this morning, so I'm going to do the presentation instead of her. I'll do my best. This time I'm going to talk about the screening we did with potential about the potential nematicidal activity of some fungus to control pine wilt disease. This work was done in collaboration with another group of the University of Valladolid, the group of Juan Pajares, but he passed away 1.5 years ago, so we had to continue the work without him, just with his group. Well, we talked the other day, the nematode, Drusapharancus xylophilus, here in Europe is considered a quarantine organism, but he doesn't work alone. He needs an insect vector, a monocamus species, and a host, a susceptible host, mainly Pinus, to develop the disease, and it all has to occur under specific environmental conditions for the disease to develop. The situation in Europe, I talked a little bit the other day also, but this is the situation. The whole Portugal is demarcated area. You can see the red color is the places where they have found nematode in trees. And in Spain we have 3 spots where we have active nematodes yet. One of them is in the south of Pontevedra. The other two are in the center west of Spain. Well, the symptoms are not specific. The susceptible trees die, but first they have the resin exudation stops, and the needles turn into brown. They have wilting of needles. There are several control measurements. We especially work, as I said the other day, in selecting resistant material, genotypes, but we also work with this group from Valladolid University, searching for some fungus capable to have any nematicidal activity against this disease, this nematode. Mainly from Stella and Boveria fungus. So, which were our objectives? We wanted to test in vitro if some fungus that we found at the Calabazanos laboratory, which is in the center of Spain, they have any nematicidal activity. They seem to have, but we want to see if it was true. Monocamus, that they have to look for a lot of samples to look for nematodes in lots of samples of monocamus, so they had some candidates. So we did an experimental in vitro experiment, but in two laboratories, one in their laboratory and another one in ours. And the second objective was to first select the best fungus in the in vitro assay, and then test them in vivo assays in plants in our greenhouse. Which were the studied fungus? Well, we chose, Stella vermicula, because there were references saying that it has a good nematicidal activity, and I think last week in the Congress in Portugal, I was not there, but I was online, and someone talked that they were having good results in the field. I don't know if it was in Japan or where, but they talk about that. We select that fungus for that reason, and then we had two different strains of Boveria bassiana, one pseudobassiana, Boveria pseudobassiana, two trichodermas, one Citrinobiridae and the other one Harthianum, and Talaromyces ruber. The design was a complete randomized complete block design with 9 treatments, because we used the 7 fungi, and then Botrytis cinerea as a positive control, and culture media as alone as a negative control. And then we work at 2 laboratories with 2 culture media, PDA and Burley, and 8 blocks. Well, first we inoculate the fungi on plates with PDA or in jars with Burley, and we maintain them at 25 degrees for in dark conditions, and then we inoculate some days later, depending on the fungus, how they grew. We have to wait a little bit or less, but then we inoculate at the same day altogether 270 nematodes in a solution of 100 microliters. And then we wait for 10 days in the same conditions, 25 degrees in the dark. After 10 days, we extract the nematodes with a Berman method, as you can see in the photographs. And the results we had were the following. First, here, this one is the Boveria, is the Botrytis cinerea, the one positive control. But, well, you can see that almost all fungus had a good result, because the population, the burst of Botrytis cinerea fungus, but, well, I will talk a little bit later about that. The Boveria species had really good results. They had no nematodes or just a few, very few number. Stella vermicula, we were surprised because we here in Lourizán, well, this is barley, no sorry, these two up are barley, these two are PDA, this is in our laboratory and this is in the other laboratory. As you can see, Stella vermicula had good, the population, the Bursaphelenchus population grew without problem in any culture media, especially in barley in our laboratory. We don't know why. And there were some small differences between laboratories, but they were not very big. Just Stella vermicula and Trichoderma citrinoviridae grew better in barley in our laboratory than in the other, and Trichoderma harthianum a little bit better in our laboratory also. But for selecting the best candidates for in vivo assay, for the in vivo assay, we analyze all data together, and the results showed, and then we select one of the Boveria bassiana. The two Boverias bassiana grew, had a good behavior. They didn't allow the population, the Bursa population to grow, so we select one Boveria bassiana and one Boveria pseudobassiana for the next assay. And Trichoderma citrinoviridae, because it has also quite good results, not so good in this one, but the median is over there. And there are references which say that it has nematocidal activity, so we wanted to test it. And then Stella vermicula, because even though we have no good results, there are people who is working with it, so we wanted to test in vivo and say what happened. The design was a randomized complete block design with 5 pre-treatments. The 4 fungus we select and PDA as a control. At 15 blocks, 2 treatments with Bursa, inoculation with Bursa or just with water, and 1 triplot. Total was 150 seedlings, 3-year-old seedlings coming from the same family with intermediate level of susceptibility. Taken with fungus, trying to not take in the PDA, but a little bit, inserting it into the tree, and then 1 month later, we inoculate the nematode 2 centimeters above the point where we had introduced the fungus. We use a 7-level scale. From 1 to 5, we consider the plant was alive, and 6 and 7, we consider the plant was dead. When the plants were dying, we try to extract all the nematodes in roots and in the aerial part. And if the plant didn't die, we extract them at the end of the assay. But the results were not so good as we expected. We didn't find differences in survival. As you can see, just 2 months after the inoculation, the nematode inoculation, almost all plants died. There were no significant differences among any fungus or the control. And when we count the nematodes in roots and stems, there were we didn't find either significant differences among them. We don't know why this happened. We think it should happen, but we have 2 options. Or I don't know, maybe, I'm sure someone here could help us. I don't know if it's the problem of the type of inoculation we did, because we took just the fungus and put it into the cat. Maybe we should have done a dilution, or I don't know how to say it, a solution with the spores, or because it was a short time lapse, because it was just 1 month since we inoculate and we inoculate the 1 month between we inoculated the fungus, and then we inoculate the nematodes. In fact, we have extra plants, and we try to recover the fungus in the plants 1 month later, 2 months later, till 8. And we found that, for instance, the Boveria, we didn't recover it until 6 months later. So maybe it's because it grows very slow. So the conclusions we have is that all the studied fungus, except Stella vermicola, had a significant negative effect on the multiplication of the nematodes in the in vitro assay compared with the positive control, the Botrytis cinerea. All Boveria had a nematicidal activity on Bursaphalangus xylophilus in vitro conditions, but we didn't have, none of the candidates fungus had a good result, a significant negative effect on the disease in the in vivo assay, probably due to those 2 causes that I have mentioned. So that's all. Thank you very much, and if you have anything that you can help, it's welcome.
Thank you. You're right on time. So could we have everyone, because we don't have David, do we? No. Could we have everyone who came to speak up to the front, and we'll have questions now. So we've got John, Martin, and are you going to do questions?
So this question is for John. John, you showed that there is clearly a quantitative relationship in resistance between white spruce and spruce budworm, but you focused on just one gene, which I'm not surprised doesn't explain very much in terms of the resistance. Have you thought about, I mean, I understand that you were focusing on characterizing genes and so on. I have other questions, but the first one is this one. The other one is the 6 forms of the gene that you highlighted. Are those is there a distribution of those within populations, across populations of Picea glauca? Sort of the population level distribution of those forms, that would be interesting.
Thanks for the questions. I'll start by answering the question about the forms. That's simpler. So these are forms that we've detected in a small number of, by sequencing and in a small number of individuals. We detect them pretty much across all of the individuals we've analyzed. But so the sequencing is carried out in just a handful of individuals. You know, the reference genotype that's used for the genome sequencing and a few others. The PCR works across quite a large spectrum, but it's PCR, so it's a bit black box. We don't know exactly what's happening. That's part of the gap that we're filling now with more targeted sequencing across populations. Regarding resistance, it is tricky to analyze what you observe in a mature tree versus what you can observe when adding these very small insects on a few branches. I don't think you can say, oh, you've only explained a little bit. We're able to observe differences. I think it's hard to say, well, how much is that really explaining? We don't know. But there is other work that showed that between resistant and susceptible trees, there are other secondary metabolites that vary, including several terpenes. We focused on this because in our transcript profiling work, the gene stood out very strongly. But there's other pathways, other metabolites that are probably quite important as well. So yes, it's perhaps a reductionist approach to things. Now we're focusing on that gene's expression, which is a phenotype. not a genotype. We don't know the molecular basis, so there may be more genes that influence the expression in addition to the copy number variation.
Thank you very much, John. I have a question for two of the speakers. I hope I'm okay, right? Yeah, this is Nadir from University of Alberta. John, we did some experiment with white spruce, looking at the different populations, about 80 different, about 400 individuals. And when we look at this, we were inspired from your work and look at those 4 acetophenones, and the concentrations of those acetophenones were really, really low compared to what you reported in Eastern Canada. So either our timing of sampling didn't work. We sampled sometime in May, sort of correspond to the budworm feeding, or there is no evolutionary significance of budworm pressure on the spruce population. That may be both. We don't know. I'm not sure if we have any comments on that. And then the second question is related to nematode. We've done a lot of work with Bavaria bassiana, basically against mountain pine beetle. We published one paper, I think, recently. There are more coming. In fact, UBC is sequencing the Bavariana genome. At the moment, I think they publish it very soon. What we found that we screened about several hundred populations of fungi, and then we found that UV is really the killer. UV lights are the killer of the fungus. You cannot use it outside. They are highly sensitive to UV lights. But there are some populations of fungi that are resistant to UV lights. You might want to focus on that aspect that may help you in your experiment, or we can talk more on that, just to give you some heads up on that. Thank you.
Do you want to respond first to the nematode question? Okay, regarding the white spruce, yes, I'm not surprised that there'll be variation across the landscape. I'm not sure which populations you looked at, but it will be interesting maybe to compare your notes with our data. Regarding the time of year, so I didn't talk about it here, but we've clearly showed that there's a clear influence of phenology. So when the foliage develops in the spring, for the first 5 to 6 weeks, the acetophenones are at quite low levels, and then they ramp up. But they stay quite stable from there on, and you can detect them in the previous year's foliage as well. So if you're in very early in the season, you might miss them. Yeah.
Thank you for the wonderful presentations, and my question is for Marcin. So my question is, in your alternating traits for the leaf phenotype, did you observe one alternating trait is more susceptible than the other?
You mean the originating cultivars? Yes, one of them is indeed the Turkey braid is considered much more resistant to rhizophora. Is that the question?
Yes, yes.
Yeah, that was one of the reasons it was used for the crosses, and it was used as the maternal component too.
Thank you.
Do we have questions on this side of the room? Or are they all over there?
Hello, I am Vilnis Šķipars from the same institute Aris Jansons is from, and I also have been working on copy number variation before, but here when you showed that the inheritance of these copy number variations is not straightforward, this let me think that it's actually a separate, very interesting direction which could be investigated because of the way how copy number variations happen, because the more of them you have, the more likely it is that something else will happen again. And we might look for some triggers for these events, and you present nice results which leave us to think about that. Thank you very much.
Yes, the inheritance results are quite intriguing. They raise more questions about selection and short-term adaptation, because things can, if the copy numbers can vary in their inheritance, that's a really interesting problem. In our recent results, we're getting indication that these genes are clustered in the genome. So if genes are clustered, you can easily imagine that there's some recombination that would dramatically affect the copy number variation. That's definitely something we want to keep our attention on.
Okay. Again, thank you for three really nice talk. And John, I think that was one of my questions, whether the genes were clustered or not. So thank you for clarifying that. That's super interesting. My other question is for Marcin. I wasn't really clear. You said that you had QTLs on linkage group 3 and 5 in your study. This other study that had a QTL on linkage group 5, I think that was your linkage group. Was that from a different material? So you basically have that QTL validated?
So you're exactly right. All three studies that investigated the recycled pulchra inheritance were based on 3 different material crosses, maternal and paternal crosses. So all the parents are completely different in all the 3 independent sites. So we are missing a lot of information, I feel like, not only because it's different starting materials, but also, as I showed you, across the years, we see so much variation with the environmental impacts on how the disease progresses and on the infection rates. So I think there is a lot to uncover. Thank you.
Any more questions?
It's actually more of a comment for Raquel. I really struggle to see the rationale for connecting the effects of what you did in vitro to what you expected in planta, right? Because you put nematodes on a lawn of mycelium in the, if I understand correctly, in the petri dishes, and then inoculated the fungi on the plants. I don't know if it's inoculation technique. I'm wondering whether, because again, it's so different to put nematodes directly on the mycelium, right, versus on the plant. I wonder if you've considered either extracting the fungi or the mycelium, or extracting the what do you call them? Fungal, not exudates, but fungal. And then applying those instead of the mycelium. So perhaps even injecting them into the trees. And then thinking about our work on systemic induced resistance, I wonder if using low aggressiveness pathogens might also induce resistance to the nematode. That might be something you may want to think about.
Thank you for the comments and the question. We consider to work with different methods of introducing the fungus inside the plant. It was supposed to be tested in the University of Valladolid, but with the problem we have with this professor, Pajares, that some of the work couldn't be done there. So it was, the project was ending, so we have to decide to do something. And we tried just I would like to compare other different methods, but you say just to try also with the extraction of the substances, I suppose it's, yes, we could try that, but our idea, we wanted to apply this, for instance, in nurseries. I don't know if we introduce the fungus, any fungus which had a nematophagous activity, if you introduce them, I don't know how, if watering a solution with the fungus, and then they would introduced inside, but I don't know because I'm not a pathologist and I don't know how to do it. But I thought that maybe that would work. But if you just apply the extracts, would it work for a long time? We don't know. Okay, because I thought that this induction of resistance maybe would work just for a few years or so, and it wouldn't work in the nurses because the nematode is going to affect the trees when they have 15 years or so. So that's why we start with this idea. Very good job. Thank you very much.
John, I have a question actually. We are currently working with acute oak decline, and we have data. We're trying to just sample either sick or healthy trees in affected sites, but some trees are in remission, so they get AOD and then they recover. We don't actually know what to do with them, the GWAS, because our GWAS is binary. So far we have been actually eliminating these trees from the data set, which is a waste. So I saw that you have susceptible and resistant trees and you did some GWAS. I was wondering how you went about this, these differences, how you made your trait quantitative if you did. Can you talk about that a little bit, please?
Okay, that's a good question. So the initial trees that I showed with dead and recovered and healthy, that was a small population that started our work. Later on, we focused more on just measuring the resistance biomarkers in our GWAS because we don't, over the last several years, even though the spruce budworm is always present in the landscape, there haven't been outbreaks in the areas of our progeny trials. So what we're measuring are the levels of the resistance biomarkers, not the level of actual resistance to the budworm, because we don't have that information. That's one of the things that's really tricky with looking at resistance, susceptibility in trees in the field. If you want to do inoculations, it's on a very small scale. GWAS has to be done on quite a large scale. And in many instances, infection in the field is quite stochastic. It's much harder to categorize. So I think that one way to set up a GWAS where you want to look at variation is when you already have a handle on phenotype that's more refined than just saying diseased versus not diseased. Does that help?
Yeah, thank you. Yeah, we have numbers of bleeds and stuff like that. We could try and come up with like an overall measure of AOD, but it's very tricky. And yeah, we don't have like 10,000 individuals like human genetics. Yeah. Do we have any more questions?
Yeah, just for John. John, I was a little bit impressed about the distribution of the species of the budworms. So, lot of species, different versions. Do you think this can be a model for this co-evolution of the gene copies and any trait or any? Yeah.
Yes, so that's the idea, is that we think that different species are applying different selection pressure, and the copy number variation would be one mechanism that allows rapid adaptation, so co-evolution, as you say. We're looking at it in Norway spruce. Obviously, Norway spruce, it's a different cohort of species. It's still very early days. We don't know if it will be a model for co-evolution of copy number variation. Excuse me. But we think that certainly there'll be some interesting results coming out of this. Yeah.
just figuring out these two models with the European very early, there's no many long time evolutionary time for co-evolving because of the migration, you know, the glacial recolonization. Instead, the American species has a much more time for co-evolve together, and
Well,
maybe,
yeah.
you know, I don't know, but
I mean, in North America, there's also been glacial expansions over the last 20 to 50,000 years. So it's also quite short term.
yeah,
But gene copy number variation, I think, allows for quite rapid evolution.
yeah, but in North America there is no the range, the mountain range are just north to south.
Yes.
So
okay.
the whole population can move up and down all together without bottlenecks and without these neutral
Okay.
processes.
I see. Yeah, that's a really good point. So we need to maybe look a bit more carefully at the at that aspect. Yes.
Because I am not a specialist in that, but it's not easy to see all those.
Yep.
More like metabolic, secondary metabolic, you know, all those different pathways, different species.
Thanks.
Do we have any more questions? Okay, so pretty much 20 minutes. Just one very silly question to Marcin, if I may. In animals, we have plasticity in color. Just wondering, can these differences in color be induced by just epigenetic factors or plasticity in itself, or are they modulated mostly by genetic control?
There are no such things as silly questions, and this is an excellent example of that. Turns out even the white flowering dogwoods have the mechanisms in them that allow them to produce color. If we put the white flowering dogwoods through the tissue culture and apply simple hormones on the media, they produce beautiful red color in the callus. So it is a matter, we feel like, and I hope that Jacqueline will prove that, it is a matter of the essentially transcription factor complexes enabling or disabling the color production. Furthermore, even in the red bracted or red-leafed dogwoods, it turns out that the temperature is bleaching the color. So there is that as well. If we have warm springs, they will start pinkish or red depending on the originality material. But if we have too high temperature, it will bleach back to white again. So it's very interesting for sure.
Okay, that's awesome. Thank you so much.
Thank you.
I think it's been 20 minutes already, so I think we can head off to the coffee break. Is that okay, Luis? Okay, awesome. Oh, thank you.
sorry, we are going to expand the coffee break up to 11. It's time for visiting the posters.
And it's my pleasure to introduce a woman with like a mother. Hello. Of the station.
Yeah, so good morning, everyone. I'm now between two institutions because I have been working in SLU as for the last 4 years, but I have recently moved to INEA, that belongs to the Spanish Research Council. So I haven't really yet any results in INEA, so I will talk about the project that payment.
So I will introduce the disease that I have been working with. I guess most of you are familiar with Phytophthora diseases. Phytophthora infestans was responsible for the potato blight that perhaps you have known before. And in forest, we have the Phytophthora cynomomae, which is quite widespread and it affects many hosts. In Sweden, we have many Phytophthora species, and in southern Sweden, where I was working, we have Phytophthora plurivora and others affecting beech and oak forest. It produces defoliation and sometimes, I think the picture is not very well. No, el puntero es este, no? But it produces like bleeding cankers in the trunk. And it's quite a big problem because it has appeared in natural parks and in places where there's the forests that we find there, they are normally like patchy. So while you see some trees that are completely asymptomatic, others are dead or they are like dying. So this is the typical landscape that we can find there. I want to introduce also a concept because it links to my presentation that is alluvion. I don't know if you are familiar with that. It was proposed by the biologist Lynn Margulis, and it's to consider the host and many other species that are living inside as one ecological unit, because they are quite linked, and we will see later how these microorganisms interact with the host. So the fungal endophytes, they are the fungi and bacteria that live inside the tissue. Normally they have a neutral or a beneficial relationship with the host. If they become detrimental, then they become pathogens. And they are becoming a potential biological tool to fight against forest diseases, and because they combine several modes of action that I will explain a bit later. So just to clarify, because I have found many times people asking what is the difference between an endophyte, or why you talk about endophytes and then you talk about microbiome. So endophytes means or refers more to like the ecology of the fungi or bacteria, and microbiome includes all the organisms that live within a host or in a particular environment. The microbiome in a forest, they are very dynamic, and they are present everywhere, from the bark to the wood. They are everywhere, and they are very diverse. They are very complex, and they establish very essential and important relationship with the host. So they are part of the ecosystem, and we cannot try to understand the ecosystem without understanding their ecology and how they interact with their host. I guess science realized how important is the microbiome when medical science found out that cancer and many other diseases of humans are related to the microbiome that we host. Like the gut microbiome is responsible for our life in time, and they are very related. So we took advantage of the technology that they were developing, and we can apply also this concept of the alluvion, like analyzing host and microbiome as one unit. But not for being small, they are less important. In fact, they are very important for their host. So why are they so? They are so important because they can help the plant to acquire nutrients. They mobilize some highly accessible nutrients They can provide like help to fix nitrogen, both bacteria and fungi that live in the roots. And they sometimes secrete some phosphorus enzymes that help the plant to capture these hard nutrients. They also promote plant growth because they produce phytohormones like auxins and gibberellins, mainly the hormones, and they increase the production of root hairs. So it allows the host to reach places where the normal roots perhaps could not arrive. And the most important, and that's why we are interested in these organisms, is because they confer protection against both abiotic stress and also against biotic stress. How they interact with pathogens inside the plant. They can parasite, the endophytes can parasite the pathogens by antibiosis because they can excrete antibiotic compounds. Perhaps the best well-known example is the penicillin that changed so much the science panorama. They compete both for space and for nutrient, and they also induce the plant-resistant mechanisms. So we can find many examples in forestry that they have been reported that microbiomes and fungal endophytes, they can limit the pathogen damage in a tropical tree and in different trees and different communities. And we have some examples of products that are based on endophytes. They can be applied to, they are mainly protective either than curative. Perhaps the best well-known example, and I guess my Scandinavian colleagues for sure they know, is the use of Phlebiopsis gigantea to fight against the Heterobasidium annosum. So while they are cutting the trees in the forest, they apply this Phlebiopsis that is a saprophyte, so it can cover the stems, so it does not allow the Heterobasidium to enter and colonize. I read that it can reduce up to 80% the colonization of Heterobasidium, so I think it's perhaps the most successful example in forestry. But we can create like a good environment for the endophytes all over or through the forest management process. So it's not, of course, we have to prepare the forest to harbor a beneficial and a good community. I know that in France, for example, when they are preparing the soil, I think it's not in forestry, but in vineyards, they do microbial amendments. So the same procedure that they adjust pH or they adjust other conditions, they can prepare the soil also for that. What we tried to find out, and our hypothesis was like in this patchy environment, in this patchy landscape that we find, besides the genetic resistance of the trees, are the microbial communities from resistant and susceptible trees different? So then, before quite tricky to get permission. So in this study, we only sampled one site. We divided the trees in 3 vitality classes: high, medium, and low. And we tried to do both the reductionist and holistic approach by both making cultures and doing whole sequencing by NGS. We So, yeah, this is me doing thousands of cultures and then grouping them into morphotypes, selecting 1 or 2 isolates per morphotype. And then we also took bark of these trees, and we extract DNA, and we did NGS with Illumina, with the ITS region. We also perform dual culture tests. So that's why it was very useful to have the collection of the endophytes. So we confront with different Phytophthora species to see how they behave in artificial conditions. And finally, we use these phenotype microarrays, I don't know if you are familiar with them, to explore how was the metabolism between Phytophthora species and endophytes. So these are 96 well plates, and there are different. We use two for carbon and one for nutrients. So when so you crush mycelia and you put a little bit there. So when you have the colorimetric reaction, it means that metabolism of the organism is activating. And if you don't have the color, it means that not. And also the intensity of the color correlates with this line. So we want to know whether if we observe reduction in growth, it was because of they use the nutrient in a more efficient way. I guess you cannot see that, but these are like the microplates that we use. You can find in the Biolog website, and these are all the compounds. There are some negative controls, so we use 2 for carbon and 1 for nitrogen, and it can be done in this Omnilog. It's like an automated machine. They are there for 96 hours. We have 4 replicate seats, and it measures the concentration every 15 minutes, which creates a huge and a monster database. So these are very preliminary results because it has been a lot of data, and I didn't have time to analyze everything. But like in brief, we obtained more than 400 isolates, like by traditional culturing, and over 900 of operational taxonomic units with Illumina. We saw that classifying trees in the vitality classes didn't create like a lot of significance here, but when we divided this classification, whether the trees had Phytophthora or not, we found most of the indexes were significant. This is all the same, but with diversity, we couldn't find here that many differences, but we did when we changed classification based on Phytophthora presence. We selected 14 endophytes that were present mainly on healthy trees or only on healthy trees, and we did this dual culture test, and we saw some of them, they present quite a reduction in the growth. This is normally Phytophthora plurivora, and this is with the confrontation. And of course, this is not like competition for space, so this one is very likely releasing some or antibiotic to the media or volatile or something that makes the reduction. This is also here. And I haven't analyzed very well this, but we see here, this is only like descriptive, but we see this is how this is Phytophthora plurivora, how it grows alone, and this is the maximum radius it has when it's confronted with all these endophytes. This is the same alone when it's with other endophytes, and this is. So we selected the ones like E2, E5, E7, E8, the ones that reduces the most the growth, and then we perform this because they are quite expensive, and we have to select the ones. So this, I think, is after some minutes. I think it's the first day. So you can see here there is no colorimetric reaction. This is Phytophthora syringae, and this is the endophyte too. So our hypothesis was that this is using glucose and other sugars in much more efficient way. So Phytophthora. Before they are very preliminary, we have to still analyze many data, is that we observe a very rich microbial diversity already in not that many samples. And we found differences in both alpha diversity and composition when we were classifying trees based on Phytophthora present and not that much on vitality status. Many fungal endophytes were able to reduce the mycelial growth in artificial conditions in plates, And we saw by the biolog microarrays that pathogens and endophytes use carbon resources and very likely nitrogen. We haven't analyzed that yet in a very different way. I want to thank Johanna Witzel for being my supervisor and friend along all these 4 years, and also all the forest pathology group in Almar. Bea knows that also belong to there, that they're a fantastic group and super nice environment. If you have a chance to visit, I strongly recommend. This is my new address in Madrid here. And lastly, I'm sorry for, I want to, Johanna asked me if I can also advertise this. This is a cost-action training school that will be held next week. And on Thursday, a company, Fenospec from the Netherlands, is coming to perform some demonstrations. So in case you can join without inscription. She told me it's free. And thank you for your attention.
Next, we have the interaction with spine.
Thank you. I'm Matilda, scientist. We all know, and I also know. So I study the interaction between one of my ongoing projects, Diplodia sapinea in Scots pine. So make the tree. So we've heard quite a few or pinea this week already. To most of you, but outbreaks of Diplodia is actually quite a new problem in Sweden. The first outbreak was discovered in 2016 by Jonas. Interestingly enough, or unfortunately, we've been contacted by several people in the past few years. They're concerned about young pine stands that are co-infected by both Melamsera and Diplodia. That's pretty cool and sad. So we want to study the antagonistic and synergistic effects between the pathogens. And the place where we do that is a site with Scots pine planted in 2015. It's the trees in the picture, and they grow fairly well, and there are no obvious signs of abiotic stress. So Melamsera pinnatorca causes pine twisting rust. It's a biotroph that alternates between Scots pine and common aspen, and it infects the annual shoots of the pine, causing them to bend or break. And we know that there is genetic variation in resistance to rust, but we have quite limited understanding of the genetic factors controlling the resistance. And Diplodia sapinea causes Diplodia piplite. It's a necrotroph, and it can sustain in or on a host without apparent disease development. And it's thought to be released from its latent phase when the tree is stressed, mostly. So the symptoms are, those trees in the picture are pretty bad, but usually you see shoot dieback, cankers, root disease, and blue stain. So it causes like substantial losses for the forest industry. And it's projected that Diplodia will cause even more severe damages with climate change. There are plenty of studies done on Diplodia saponaea, especially in Austrian pine. So we know that the fungus benefits from the tree's response to stress, especially drought and hail damage. We also know that the disease development is affected by the microbiome in the tree, and in general, necrotrophs can exploit the tree's response to biotrophic. Melanthera predisposes the trees to Diplodia tiplite. And we think that the disease severity is explained either by the stress metabolite that is produced by the tree or the tree's microbiome. So we have surveyed the symptoms of pine twisting rust and Diplodia tiplite in subsequent years and sampled buds from the trees and spores from the fungus to study the population genetics. We've also sampled RNA for study of the gene expression in infected tissue and shoots to study the metabolites and the fungal community. And we also measured all the trees and scored the presence of bifurcation. I'm really bad with keeping the mic where it should be, I'm sorry. So after the first damage estimation, it was quite clear that Melanthera infections do not explain Diplodia tiplite, because we have about 575 trees included in this study, and I didn't find one single tree without Melanthera symptoms, and very few trees with a low level of infections. So the top figure here shows the tree vigor, which is just the score I gave the tree, if it looks fine, if it's ugly but probably fine, or if it looks really bad. And as you can see in the top plot, the Melanthera ratio is quite high in most of the trees, but many of them still look fine. So the bottom plot shows the number of Diplodia infected shoots. And the trees look worse. I have not done any statistical analysis on this, but the trees look worse if they have more Diplodia infections. So the tree to the left has Melanthera infections in about 70% of its shoots and no Diplodia, and it looks fine. The tree to the right has Melanthera in about 85% of its shoots and a lot of Diplodia. And I classified that as a bad one. So the sampling for metabolite profiling and fungal community was done on trees in 3 groups, and the groups were based on the symptoms of the trees in the previous year. So we looked at healthy trees. There were no really healthy trees, but low levels of Melanthera and no Diplodia. Trees with a lot of Melanthera but no Diplodia, and trees with a lot of symptoms from both pathogens. And the same trees were sampled at 3 times. And the first time point is to the left, beginning of June, when we could see Melanthera infections appearing on the shoots. It's a bit dark, but it's the yellow stains on the shoots to the left. The second time point was a week later when Melanthera was sporulating. And the third time point was a month after that when Melanthera seemed to no longer be active and the trees began to heal. And we sampled asymptomatic and symptomatic shoots from all the trees. So after processing the shoots, we divided all the samples into 2. So one part for fungal community study, and one part was sent to our collaborators at Max Planck Institute for, they analyzed the amino acids and phenolic compounds. So the amino acids didn't show anything interesting. There was a large variation both among the groups of trees and the symptoms of the shoots. But this NMDS shows the phenolics, and the different colors are at the different time points. And as you can see, there's quite a clear separation between the time points. And there's also a very clear separation between symptomatic shoots and asymptomatic shoots in the last time point. So the symptomatic shoots are the field points. I love that I'm pointing, but I'm not using the pointer. But the asymptomatic shoots are the empty points. There's still a lot of variation among the groups, so the previous year's symptoms do not explain the phenolic profile. I don't know exactly what it means, but I think it's interesting. There are, of course, a lot of analysis to do still. I've told you that I'm going to analyze the fungal community. We want to do a lot of what Carmen has done. So I'll get to that. And I would also like to understand the stress caused by Melamsra better, because just counting the infections don't seem to tell us anything about how the tree is doing. So maybe the models where I include Melamsra symptoms in subsequent years and include the growth for several years. And then, of course, the most interesting part, the genetics of the trees. I don't know if Malin maybe already sent the order for genotyping of our samples, but yeah, it's on its way. Yeah, so I look forward to tell you more when I have some real results. That's all from me. I'm not alone working on this project, so I want to thank my supervisors. They're very cute. My field assistants, that's Malin and Janne to the left. I guess you understood that. Sebastian, David, and Maria has helped me in the field, and my dog has supervised a lot. Oh, and I forgot about the funding. Brattås Stiftelsen, Carl Tryggers, and the Forest Damage Center at SLU has funded this. Okay, thank you for listening.
Hola. So next speaker is Fan Long, that will talk about tree microbiome of European ash and is shaped by environmental factors. So.
Good morning, everyone. My name is Phong Long, and I am a PhD student from University of Copenhagen. And today I'm going to talk about tree microbiome of European ash, which everyone already know which it is, shaped by environmental factors. So the European ash, which is medium-sized deciduous tree, and it is wind pollinated, and also it's growing throughout most of European countries. Like this map shows, except for part of the Norway and Finland and the Sweden, and also most of the Spain, they don't have much this ash. Yeah. So now in Europe, the main threat is ash tabac, which I think most of people also know this. It is caused by Hymenocipherus fraxinus, and it is an invasive pathogen from Asia, and the first observed was in Poland in 1992, and the first observed in Denmark was in 2002. And it is also spread to the most of European countries, and which can cause symptoms and on the majority of trees, and also the cause severe mortality. We can see from here the adult tree, the symptom, and also the symptom from the small seedling, the brown dot here. But however, now there are still a small fraction of the ash tree that remain healthy and also minimum symptoms. So in our previous study, the results showed differences in fungal community composition between healthy and unhealthy ash trees. In this study, we would like to confirm this pattern. In Denmark, we have two sister trials, which is here, Nandes, and also Silkeborg. These two trials was established in 2004, and we can see from here, this is the overview of the Nandes trial. We can see here there's a good-looking tree that looks healthy, and also a lot of symptom tree here, or almost that one. And also this is the one look very healthy in Silkeborg trial. So for this study, the hypothesis is there is a relationship between susceptibility to ash dieback and the fungal community. The second one is fungal community is influenced by site. And the third one is fungal community is influenced by a metallo effect. So this is our workflow. We collect 15 families from both sides, and for each family we collect healthy and also unhealthy trees. In total, we collected 108 twig samples. And because we studied about the endophytic fungal community, so we did the surface sterilization, and then we do the extraction and PCR, and also the ITS1 applicant sequencing, and then the bioinformatics analysis. Here, this is the one of my colleague, James. He helped me collect the twig samples. Yeah. I will show the preliminary results, what I have got so far. The whole samples, we got 161 OTUS, and also there are 81% of OTUS were present at both sites. And for all of this OTU, we have 110 OTUS belong to ascomycetes, and 36 OTU belong to basiomycetes, and there are 15 OTUS, we don't know what it is. And also there's no OTU was observed to belong to other phyla of the fungal kingdom. Yeah, and this is the composition of the fungal community. This one is the class, taxa class. We found here, from here we can see the dominated fungal taxi. It is within two classes, which is Docidiomycetes. It is the green one, and also the Uratiomycetes, which is the orange one. We can see they have a large portion of the community. And also this one is the fungal genus in among the 15 families. As we can see here, we have 3 of the general. They were among the prevalent general across these 15 families. Oh, the color is not that classification, yeah, but yeah, it is. So we also did the composition analysis among the health levels. So which is the 0 is no symptoms, and the A and B, C, D is have some symptoms, and also this one is almost dead. So from here we also have the kind of same result like before. We have the two dominated classes and also dominated general. And but as we can see here, this one we can see the unidentified is like a lot of unidentified, but it's because it could because we only have one tree. We only have one tree collected from the site, and so here we can see it's a little bit, yeah. And this is also the general for the different health level. We also can get the same result like among the different families. So for the alpha diversity, we analyzed we got the observed OTUs and also the Chow-1, the richness, and also both of these two, they have significant influence between sites. However, the Shannon diversity, there's no significant difference between sites. The alpha diversity among different families, which is a little bit sad, we didn't find significant difference among the 15 families of all like observed OTU, Chow-1 or Shannon. The same result was from the health levels. We also didn't find the significant difference among different health levels. So for the beta diversity, we compared these two sites. The PCOE figure shows the differentiation between these two sites. We can see from here the green dot is Nandos, the Silkeborg is the red triangle. We can see they have a little differentiation from here. And from the Permanova result, we see the site has had a significant influence on this fungal community structure. And also, there is like here, there's a interaction between site and also the health level. They also have significant influence of the to the fungal community. We also did the dispersion test, which is also significant. This is also a little bit interesting, so we will look at it further. So this is my preliminary conclusions for the first hypothesis. The answer is no, but we found a significant interaction between the health level and site. The second one, yes, but the homogeneity test was also significant. For the third one, the answer is no. We didn't find the significant influence of the metallo effect. And I also, for this study, I would like to thank to my supervisors, which is Nina Roscoe and also Eric and Chachai and James. And also, I would like to thank the support from the SNS and the Norwegian, and also the project grant from Hartman, and also the Chinese CSC grant. Thank you.
To the last speaker of the session. An epidemic in Scots mine.
by Janne, and project. So I would not like to say I take ownership of everything here. I will try to explain it to the best of my abilities, but they only stretch so far. So that's all the disclaimers I'm going to give. So basically, let's move. Let's tell you who actually did the work first. And there are a couple of people that needs to be mentioned here. Berit Samuels and Juha Kaitera. They are the specialists on Cronartium rusts in the northern Scandinavia. Torgny, now that's moving. What happened now? Now, here we are, yes. Wrong button. So Torgny Persson and Henrik Svennerstam are the specialists on the tree species on Scots pine in the north. Torgny is the breeder and Henrik is the sort of management specialist. And then we had a massive support from various industry and sort of agencies in Sweden to deal with this problem. And over the last 20 years, or roughly 20 years ago, we started to have signals coming in from the industry that their pine regeneration kind of failed. So this picture is actually roughly 20 years ago. And as you see in this picture, there is a lot of royally unhealthy pines here. And this, in the long run, will lead to that this stand is understocked. And if this continues, this also means that the owner will have to take it down too early and start over. And the major fungal culprit for this situation is Cronartium pini, spruce Scots pine blister rust. And we've seen a lot of blister rust pictures, and this is no different. We will have these dioecial spore blisters formed on the pine. We also have kind of swellings where the disease is progressing, and then on the edges of the swelling you will find spore blisters. Eventually, needs to be said, eventually this disease starts on the needles, grows into the twig, and eventually it will reach the main stem. Then it will destroy the vascular system in the main stem, and you have this phenotype that the tissues above the infection dies, leaving the trees either dying or severely bifurcated, which is not good. And for those of you who are interested in sort of the epidemic as such, I recommend this paper. So Cronartium pini has, it's a rust, it has a full heteroaceous life cycle, where pine is the acial host, and various herbs in the forest is the teleal host. And I say various herbs, I truly mean here is a lot of herbs, because this pathogen is present throughout the whole of Europe and Asia, so it will also have various pines that it's able to infect. In Scandinavia, we also find, this is really not good now with managing the mic here, a sort of truncated cycle, an autoacious cycle. So we have both, and we have them present in the country. And if we look at the populations of the heteroaceous and the autoaceous forms of this rust, we actually have multiple populations of both, but they are distinct. So the next thing we can see is also that we have no clear geographic pattern where we have the autoaceous and where we have the heteroaceous form present. So it's not to us really, really clear if these different forms, well, how they interact with the host and how that drives the disease, but we have some hints to it, as you will see in subsequent slides. So to give you an idea of the magnitude of the problem, we have roughly 80,000 hectares of young pine stands in the age from 5 to 30 years, which are severely understocked. And 42,000 of these hectares are present in the northernmost part of the country, the northernmost coastal region part of the country. I put a little box up here so you have an idea of which half of Sweden we are in. So that is a pretty large chunk of the suitable pine land which is gone, and it's in total 20% of the forest land in that area. So it's bad. But if we then look at the heteroecious and autoecious form of the rust, and we're starting to get clues to what's going on. So the left panel here, we have the heteroecious form marked up with green pegs, and the autoecious form marked up with orange pegs. And in the left, from your end, right panel, there is also the disease severity indices. And if the stand is marked with a red dot, that means it's severely affected. And then you can see that we're starting to see a pattern that where we have the severely affected stands, we also have the heteroecious form present. So we would assume that, based on this data, that when we have the alternating rust present, we have more problems. But we don't at this point have a good idea of why. Are there differences in infection efficacy when you move from one host to the other, or are there differences in aggressiveness between the two forms? Another aspect of this inventory of the severity of the disease is that we also can identify some silvicultural risk factors, such as the presence of the alternative host, Melampyrium sylvaticum, and I mean no prize for guessing. If you have problems with the heteroecious form and you have the Melampyrium present, yep, sure. It's also sites with very high growth potential, so the trees would grow really well without a problem. And it's generally sites which a forester should have most likely have planted spruce if they would have sort of just looked at the site properties. So there is also silvicultural practices involved in this. So if we move on to sort of my favorite bit of this, and that's of course the genetic key parameters, are the things we can do by using the genetic resources. And I would argue that the case to this is, the answer to that question is yes, there are things we can do, because if you look at the graph to the right, there is a substantial degree of variation in the northern Scots pine population for this trait, meaning how susceptible it is to the rust. Needs to be said also that Scots pine is, to start with, not the most susceptible pine species to this rust. So the heritability values are in the order of 0.4, which is a lot similar to the values we have for growth, for instance, which we can breed for rather successfully. So if we know how to identify the trees and how, well, then we probably can breed. So this is where we are currently trying to devise strategies for doing so, finding a way to identify the trees early on, which have a good resistance profile. And we have basically chosen 2 different strategies for this. How am I for time, Carmen? How many minutes? 6? Okay, good. So one is based on the concept of breeding without breeding, and that's basically a concept where you go out in a stand, you phenotype the standing variation, and then you also genotype these individuals and their parents. And then using that, you do pedigree reconstruction. And in that way, you can create sort of an idea, a sort of virtual progeny test. And the other way forward is, of course, to do what's been done rather successfully in North America, to attempt to build a screening center of some kind. And I'm going to give you a status update on where we are with these 2 strategies. So in Sweden, and I think also in Finland, for instance, where we have this problem, the majority of the stands are, after clear cutting, replanted with nursery material that originates from various seed orchards. That's sort of making up the deployment population from the breeding program. And our thinking is that this is information we can use to go back up and see if we have put unsuitable clones into the seed orchards and whether those clones should remain. by applying a breeding without breeding approach. So we use then these really heavily affected sites in the north. We have subsampled some of these places, collected DNA from these, and then we have also collected DNAs from their from their maternal trees, which are located in a seed orchard outside Umeå in northern Sweden. And then and we know that there are trees in this orchard which isn't exactly perfect in terms of rust resistance, but maybe we haven't caught them all. So what we then did was we phenotyped these trees, we collected the DNA, and we are now in the process of genotyping these individuals. So this is pretty much also the strategy that we will use for Matilda site, that we will genotype these individuals and their mothers, because also that site is derived from a specific seed orchard, though a different one. And then the other strategy is, of course, the the screening center, and this is work from Berit Samuels and Torgny Persson. And they have collected spores from Scots pine in the forest, and then inoculated these spores on peonies, which is a alternative host for Scots pine blister rust, and achieved to allow the fungus to create basidiospores, which means that they can use basidiospore drop inoculation techniques to inoculate seedlings, which is what's shown here on the picture in the middle. And in addition to that, they've also collected spores from the autoecious form and sprayed that onto Scots pine seedlings of various ages. This is a pilot study that was carried out this summer, so all the data we have at this stage is very, very preliminary. So basically, after spraying with the different forms or allowing the spores to drop on the needles, we do actually see chlorosis or necrosis on the needles, suggesting that at least the needles gets infected. And diagnostic PCR also validate that these lesions does contain the fungus. So basically, at this stage, we have managed to get the fungus into the plant. And this is where we are. Now these seedlings will have to be followed over time to be comparing then whether we see a difference between the susceptible and the resistant proteins from the susceptible and resistant mother trees. So if I'm coming to a conclusion on this talk, there are some points that we want to make about the Scots pine blister rust problem in the northern part of Scandinavia, and that it's not a new problem. It's an old problem. It's a native disease that have started to bloom very recently, and we don't really know why, but we have the leads are that there might be changes in the forestry practice. We're planting the wrong tree at the wrong site. That's a standard. We have perhaps changed which host genotypes that we are putting out in the different sites. And we know that the climate is changing. We know that this rust disease is favored by warmer climate and wetter climate. And in the north of Scandinavia, this is what we are going to get, warmer and wetter. And the last 30 years have been much warmer and much wetter than previously. Then we have the sort of black box, which is what does the autoecious and the heteroecious form of the rust do, and the host genetics. And this is what we need to follow up, setting up a screening center and by following up these two organisms' genomes and population genetics. And by this, I will thank you for listening. Thank you.
Any questions?
Yes, I had a question for Malin regarding the breeding without breeding approach. I think it's really interesting as an approach because it allows you to potentially carry out genetic studies without having pre-established field trials, and with changing conditions it gives us a lot of flexibility. I'd like to get your idea of how, do you think this will work well? Are there any challenges? Is it being kind of seen as a really promising avenue for genetics and genomics in Sweden? Just general impressions.
I think that currently it's maybe getting a little too much attention in Sweden in relation to how good the method will be. It's currently been seen as, well, now we have a cheap and efficient way of doing this. And we really don't have a good proof of concept yet, and this is also why I had the line of one segregant analysis up on the slide, even if I didn't talk about it, because I haven't seen that we can reconstruct the pedigrees yet, because that's really, really crucial. Because when the sites looks like in the first picture, they could be volunteers, and that's going to make it quite interesting. I think it will work, but not for every site and every population. So it's not a super quick fix, but it's going to be helpful.
Yeah, well, good morning. I have maybe a comment and a question. And the comment is that I know in Norway there's a lot of trouble with that spruce is planted on pine sites, but I never heard about planting pine on spruce sites. So I guess since Nordic forestry is in love with spruce, that would be an easy solution, just to switch over to spruce, and it will keep healthy for maybe the next 100 years at that northern latitude.
I was surprised too.
Yeah, and then maybe disclaimer is that I'm not a geneticist, but since you have this the host switching, sexually reproducing pathogen population, it will, I assume that it will be more genetically variable, so that might or somehow be part of the explanation that you see much more severe symptoms, perhaps.
I think so too. I mean, the thing is with the autologous form is if that's a good clone, which is good and has sort of competitive capacities, then it can dominate very nicely. So that's why that's probably there. But I agree with you that I think that the host alternation there is altering variant have some kind of fitness advantage. We just don't understand what it is.
It's Marlin. When you compare the heterocious Acronaton pinna with that of the autoecious Acronaton pinna, I don't know if you mentioned it and I missed it, but what's the difference in terms of their life cycle? Like, how long does it differ in years or?
You mean if we compare the heteroecious form and the autoecious form?
Yeah.
Without molecular markers, we can't differentiate them. It's really, they are as I mean, there is no way when you stand in the forest to say this is either A or B. It's you bring, so they seem to be, from my naive, and this question you should pose to Berit Samuels, from my naive understanding of the whole thing, they seem to be rather persistent, both of them. Did I answer your question?
No, what I'm saying is, let's say, for instance, the heterocious form takes like 4 years to start producing spores, and then the autoecious form, how long does it take? Is it 2-3 years?
No, but that moves from pine to pine within the season.
Okay. All right.
My question is about the microbiome studies, or well, both of you guys, because it seems to me your basic hypothesis is that different microbiomes or microbiomes will confer different resistances to the trees. But the way the studies are set up, it's like a chicken and egg situation, right? Because you're looking at diseased trees, you don't know if the pathogen is modifying the microbiome or if the microbiome is affecting the pathogen. So do you see a way out of this to actually test the hypothesis?
What I thought is between the healthy and unhealthy, and also the unhealthy one, there's a pathogen in the tree, and there could be have the interaction between the pathogen and the microbiome of the tree. So basically, I think they can maybe they have like what is called yes, suppression. The pathogen grows in the tree. So I understand your question properly. So I think it's very tricky. What we have tried to do is like using clones in different environments to see like, so effect of environment is very powerful. This is we know. If we get rid of this environment like effect, how can we determine if resistant trees harbor different communities, or if the chemical ecology of the resistant creates a niche for that? We have tried to create completely sterile seedlings. It's a bit tricky because they inherit a little bit from the mothers, so they're not completely sterile. So I know there is a group in the US, I think it's in Oregon University, that they are creating to inoculate these synthetic communities, to see like first like the order of arrival, which is called priority effect. They know it has like a lot of power, and then as you say, is it like the resistance of the tree that is created a different niche to overcome. So, but I think it's very tricky, and I have also the same feeling that I don't know. I'm not like looking at the whole picture because it's very difficult to establish what was first, because I think it's very difficult to create like the order of arrival. And I know also that they have analyzed the sequences, so to establish like evolutionary relationships among the fungal communities. But yeah, I'm not microbial ecologist, so I cannot answer, but I understand, and I have also wondered that.
Yeah, I have a suggestion. Maybe we can follow up later, but I tried to get a grant to work on something like this with sudden oak death in California, right? Because we actually have a screening tool, which is our IR-based screening, to predict the resistance of trees before they become infected. And we wanted to compare the microbiomes of those trees and then challenge the trees with the pathogen, do artificial inoculations. Well, we didn't get the funding, but maybe that's one way to do it, right? If you could predict which tree is resistant and which one is susceptible, then you can maybe you can answer that question, right?
What we have compared in a common garden experiment with different pine species is that the pine species, even regardless the resistance or susceptibility, this was a study with Fusarium delphinatum, the pine species was the most important factor shaping the community, regardless that the plants were planted and produced in the same greenhouse and whatever. So yeah, the chemical ecology of the tree, of course, is making a lot. So.
Yeah, I have a question to Matilda, and the question is Melamsora pinitorca, has it a similar pattern as the Corynatium that we've seen? So is it a re-emerging disease in Sweden, or is it just popping up, or is it just consistently through all the time, have been the last 20, 30 years?
It's been
are increasing, but it's a native species. But I also think that, like, as I've seen, that it usually doesn't kill the trees, stunts the growth.
And another question is, do they reproduce sexually? So you have an oocytes populus tremula?
Sorry,
do you have populus tremula on oocytes that are affected by, yeah, okay.
It's me. Carmen, this is a question for you. I really appreciate it for providing really nice overview of microbial symbiont. I learned a lot. I am new in that field. One of your results, I think, was very fascinating to me. You mentioned that based on your microarray work, that you say pathogens and endophytes used carbon resource in a different way. Could you please open that one? I don't know what do you mean?
So our hypothesis was that, and it was not only our hypothesis. Like, we have read before, like trichoderma, for example, that is being used in some nurseries as biological control agent, is very efficient transporting glucose. So since it's so efficient that it can take advantage of other organisms. So when there is limited resources, and if they transport glucose in a very efficient way, they grow very quickly, and so the mode of action of this biological control agent is like a competition for resources. So if they're very efficient on that, yeah, it's true that it does not work like that always, because the pathogens are bullying sometimes, and even though the others, they are more efficient, they sometimes can change that. But yeah, I didn't want to go through more, but this is like general explanation.
Have you tried plant secondary compounds, your microarray work, phytopathogens versus endophytes? Which one going to be successful for growing, depending on the media? But media is based on the plant secondary compounds. Instead of sugar re-adding, if you added the plant secondary compounds, which one do you think is going to be more successful for growing on that kind of media? Highly toxic, I assume.
Which endophyte you mean, or?
Endophytes versus pathogens. I mean, endophytes can utilize the plant secondary compound as a carbon source. That's why I'm after, basically. What's your opinion on that?
On the plant, you mean?
Yeah.
Well, I did my PhD on that. And I don't know if I can answer. Yeah, we got nice results on trichodermas, even though in the plants they were growing very fast. So they were able to reduce necrosis even in plants, and they were able also to induce the plant immune system also. We only measure total phenols and terpenoids, but they were able to induce that. So yeah, sometimes it's not only one mode of action that they have, but they have several, and they employ several. So that's why they are successful in sometimes. I know trichoderma, they are being used in nurseries. I know that one strain of verticillum is injected in elms also as a way, but yeah, the thing is how long this lasts, how many times you have to apply. I know 1 year, but it depends on the organisms, and it's quite complex, I guess.
Jeff, thank you for your patience.
Yeah, this was a great series of talks. I have a lot of questions, and I'm not going to ask any of them really, but so it's hard to decide which questions to ask. But I would love to ask some questions to Feng and Carmen about the microbiome and its interaction with disease. And I think you did a really good job of setting up just how complex and difficult it can be to interpret and design experiments to learn things. And I think that Enrico and Nadir really did a good job of also kind of highlighting how do we pick apart, cause and effect, or interpreting these pairwise competition assays on basic media, or maybe how would, including other substrates or conditions in those assays
maybe improve the utility of these assays and understanding all these interactions, right? And I actually wanted to just make a comment about specifically the, well, first of all, the question, well, it's not a question. I mean, I think it's really important to acknowledge this, because we've also seen it's difficult to like tease this apart, to design experiments to tease apart what's the role of the environment, what's the role of the genetics, what's the role of all of these individual components of the microbiome. And there's so many studies on the microbiome, and it's like, what have we learned and what have we not learned, right? And so actually, I was getting to it though. So in the Permanova, you have factors and interactions that you were looking at. And I wanted to point out that I know that your major takeaway maybe had more to do with another thing. But I wanted to point out that you had also R² values in that, right? So you can look at the p-values, and maybe some of them are not below 0.05. But you had something really interesting in there, which was genetics by the severity of disease, or the presence of disease. And that was your highest R² value. It was like something like 0.09 or something like that. But I mean, going back to Enrico's question, we do know that from many studies that pathogen infection changes the microbiome. So we know that, right? What we don't know is there some sort of thing that we can predict or understand about the microbiome that we can expect to influence disease? That's what we're really interested in. But I think that that's something that you should pay attention to in that Permanova, because that was the highest amount of variation you were accounting for, was that interaction between genetics and the disease on the microbiome. That's all I wanted to say. Sorry it took me so long.
Yeah, just I have I'm not a specialist, but I love all these mutualistic or not negative interactions between organisms and plants, and just asking to you for what we know. We know that endophytes, or not parasites. No, I mean not antagonists. Fungi can change the phenotype of the plant. They can change even gene expression, metabolic pathways, whatever. So at what level there is evidences that the endophytes change the metabolisms? It's just expression of enzymes or control over enzymes. They are changing resources. They are changing maybe the expression at the nuclear level, or they are also inducing epigenetic marks that can be inherited in the somatic line of the growing tree.
Yeah, we were just discussing something similar, Pauline and I, yesterday, I think, because I don't know. This is like the expert answer, but I read and I know that sometimes. They can change the environment where they live. This is true, and they can change by expressing their genetic material in a way. I'm talking about microbes, and they can change the chemical environment that they have also. I don't know much more in detail, but I know they have quite powerful tools to change and the way that they interact.
I'm not sure I can follow, but I can, but I'm not sure I can follow up on that one very easily. The first point I would like to make there, Luis, is that if you look at symbiotic fungi like mycorrhizal fungi, which is something we don't discuss at this conference, they have effectors too. They manipulate their environment too. I think that many fungi manipulate their environment that they're in, and they will do it towards their host, the tree, but they will also do it again to the surrounding microbiome. And what Carmen was alluding to is that there are studies where people have followed where, and I'm trying to explain now, it's not only proteinaceous effectors which is manipulating the host or the surrounding surrounding microflora. It is also uptake of microbial RNA into host cells, which is going to influence the host cell. It's also uptake of sometimes actually microbial RNA into other microbes, which is then manipulating those. And I think this is a factor which is extremely difficult to research, and especially in our systems, which is slightly perennial, to say something, for the host trees. But I think that's a very important aspect of what is going on, and I think Carmen is right when she says this is important, but very difficult.
So, I mean this. About I'm interested a little bit more in this epigenetic marks, maybe methylation of the host that can be inherited. So, you know, if there is any evidences that, but if this is happen when a cell growing at the end of a branch, or endophyte reach that cell, they can modify the expression for longer time than more than one generation. So it can be inherited along that branch. So that branch can be, you know, different than the other and whatever. And so the phenotype inside the tree is going to be different, that side, the other. And
I'm not sure evidence, because, or at least, let's say, I'm very poorly read. I haven't read very much literature in that domain, but I'm kind of Team Honasoliv on that one, with the term of maternal provisioning also, that it doesn't necessarily need to be genetic imprinting. And also when it comes to genetic imprinting, then maybe Paul should be commenting, because the Norwegians are doing a lot of work on that one. But that also it's a matter of provisioning, that when you are, whatever environment the meiosis takes place in, and then the fertilization and the production of the zygote and the endosperm, or megagametophite, that's going to influence what happens in the next generation. So it doesn't have to, in my opinion, to be epigenetic, even if there is definitely epigenetic effects as well. There's a lot of things going on during sexual reproduction.
It seems like we have a lot of interest, so I may suggest maybe additional discussion on this, but let's finish with one quick question from Marcin, very specific. And
yes,
then one
it is fast question, and Malin, you may keep the microphone. It's to you, please. All talks were great, but yours stood out definitely, in my opinion. And my question is, if you have this wonderful setup for coevolution between the host and the parasite, would you think with a multi-cycle behavior of the parasite, you could actually be screening for resistance or putting the pressure for resistance if you put the good clones somewhere in the longer term, that the parasite would be actually speeding up even faster?
You mean experimental evolution? Yeah, if we have the inoculation system up and running and steady funding, because it will take, as the people with the screening centers in the US say, it's not a quick thing to go from one stage to the next stage. And usually it would keel over on the long-term funding, I think. But in theory, absolutely.
A quick follow-up on Luis's questions and comments. There's a lot of information on endophytes in grasses that show that the endophytes themselves either produce chemicals, antibiotics or whatever, and thereby that almost certainly will affect the metabolism of the host. And then there's a whole thing about vertical transmission, right? So you can have the same endophyte in the mother plant that will end up in the seed, and therefore the progeny. So that effect potentially can be perpetuated in that way. Just a couple of comments.
Please join me in thanking everybody, especially our speakers. And I think
okay, so with this, we finish the session of the morning. We have any question about this logistics or whatever? No. Okay, the lunch time is at 1. we have some time now for watching at looking at the posters, questions, and if you want, we can walk around a little bit. There is a nice visit that I can